Hey folks, I think we'll get started. So hello everyone, my name is Megan Mahoney and I'm the Director of Scientific Affairs and Training Programs at BioCanRx. And I'm very pleased to welcome you to the second session of our BioCanRx Summit Speaker Series. This session and this series were developed because our face-to-face summit for Cancer Immunotherapy conference was postponed to 2021. And we thought this would be a good opportunity to bring our network together and share the data, the research from our funded network investigators. And so in today's session, we'll be talking about developing better CAR T therapies by engaging patients, performing systematic reviews, and assessing real-world and economic evidence. For more information on the schedule for today's session, as well as the speaker bios, please visit our website that is linked in the chat box. So, like session one, like session one, we will be starting with a plain language summary by Madison Foster, who's a clinical research assistant with at OHRI, working with doctors Manoj Lalu and Dean Fergusson. I'll hand that off in a moment. But before I do that, I'd like to remind everyone that we will be holding a third session that will be taking place in January in 2021. And so I would recommend checking at the link that's provided in the website and also following us on social media. So at this point, I would like to hand it over to Madison. So Madison will be speaking first, and then we'll have Terry Harwash, who will be presenting a patient perspective. And so at that point, Madison, I'd hand it over to you to share your slides.
Okay, great. Can everyone see the slides okay? Okay, wonderful. So thanks so much, Megan. So as mentioned today, I'll just be sharing a brief talk or a plain language summary on some of the topics that we're going to be hearing more about this afternoon, which really focus on developing better CAR T-cell therapies by engaging patients, performing systematic reviews, and assessing real-world and economic evidence. And so I'm just going to provide you with my perspective as a research assistant, and then I'm going to hand things over to Terry, who's going to provide you with his perspective as well. Okay, great. And so we also just wanted to start off by noting that the research projects that you're going to hear about today, they really involve quite large and collaborative research teams. So we're not going to be able to hear from everyone, of course, but we will be hearing from a number of the lead researchers or investigators, and as well a couple of the HQP or research assistants or research associates involved in the projects. And so just to note that the first 4 talks that we're going to be hearing about this afternoon by Dr. Fergusson, Dr. Thavorn, Mackenzie, and Gisell, these will be focused on sub-projects that are part of a much larger research program, which was nicknamed GoCart. And so I'll provide a little bit of background on that research program. And then the final 2 talks that we'll be hearing about from Joshua Montroy and Dr. Manoj Lalu, these will focus on 2 newer projects that are just starting up in 2020 and which really build on some of the work that was, some of the results and findings from the GoCart research program. And then at the end, we'll have a question and answer session moderated by Dr. Justin Presseau. So just to give you a little bit more background on some of the research projects that we'll be hearing about this afternoon. Really, the overall research goal or objective of these projects was to develop a clinical trial protocol or to design a clinical trial. And this was for an early phase trial or a phase 1/2 trial, which just means that it's aiming to test the safety and feasibility of the therapy. And so specifically, this was for a Chimeric Antigen Receptor T-cell or CAR T therapy for relapsed or refractory blood cancers. And so just for those who might not be familiar with this type of therapy, this is a personalized immunotherapy, which actually uses a patient's own immune cells, which can be collected from a large blood draw. And then these cells are modified genetically within the lab so that they're able to identify and attack cancer cells once they're given back to the patient. And so really the talks that you're going to be hearing about this afternoon, they're going to describe kind of the new approach that the research team took to designing the clinical trial, and just a little bit more about the rationale behind why this approach was taken. Really, with a clinical trial, this is no different from any other project where you might be embarking on a project or the trial, and you have this very clear path in your mind of how you're going to complete the project. And it looks like it's going to be relatively smooth sailing. But then in reality, we know that certain barriers or hurdles might kind of pop up along the way. And so the team wanted to take a little bit of a new approach to designing the clinical trial. And they took a team science approach where they involved researchers and doctors from a variety of different research areas, so that the team could work to compile evidence from each of these different research areas and combine this to build a really rigorous clinical trial protocol. And as well, another large component of the research program involved engaging patients throughout this development process. And so the idea was to engage patients and involve patient perspectives throughout the development of the clinical trial. And overall, the hope was that we would be able to identify what some of these barriers or hurdles were well in advance, so that when the team was designing the clinical trial protocol, they could hopefully work to address or reduce barriers or build in the necessary supports for trial participants, researchers, and the doctors involved in the trial, and hopefully better align reality with expectation. And so my next slide just shares a little bit more detail about the approach that the research team took. So as mentioned, the larger research program was nicknamed GO-CART for Getting Better Outcomes with CAR T-Cell Therapy, and it was aiming to develop the clinical trial protocol for a CAR T-cell clinical trial, the CLIC-01 trial. And the team nicknamed their approach or framework or model the Excelerator, as it aims to accelerate discoveries to a trial and hopefully eventually one day the clinic or the bedside. And so this approach involves a couple of different studies. And so I'm just going to walk you through them one at a time. But just to note that at the same time, the team was also taking an integrated knowledge translation approach, where we worked with patient partners and engaged them throughout the research process through meaningful collaboration, where they were actively engaged throughout some of the research activities. But I won't be going into too much detail on that side of things, as I'm going to pass things over to Terry afterwards to tell you a little more about that. So the first project involved in the Excelerator program is the systematic review, which involves identifying existing preclinical or clinical data, so animal studies or previously completed clinical trials, so that we can compile this data and pull it together to take a look at what existing data there already is on CAR T-cell therapy for blood cancers. The next core project within the Excelerator framework is the implementation diagnostic studies. And so this involved interviews and surveys with both patients and hematologists to get a better idea of what might be potential barriers to participating in the clinical trial, and as well what might be helpful supports or enablers throughout the trial. And so we're going to be hearing more from Gisell about this core project. The next study involved in this framework was the early economic analysis. And so this part of the project involved using economic models to take a look at factors that might influence or help to maximize the economic feasibility of CAR T-cell therapy in Canada, so that this could be considered when designing the clinical trial. And so we're going to be hearing more from Dr. Kednapa Thavorn about this project, and as well from Mackenzie Wilson on how they're engaging stakeholders throughout this project. The next core component study was a retrospective cohort study, which just means that the team took a look at previously collected hospital data to kind of test out the eligibility criteria, just to make sure that it was going to be feasible and see if any changes were needed for the trial itself. And the last project involved testing and validation of the therapy, which was done by our team members and collaborators in Vancouver. And so we won't be hearing more about these last two core projects today, but just to note that they are a part of the overall Excelerator framework. And so after the team had gone through each of these studies, they worked to develop the clinical trial protocol. And as Terry is going to tell you a little bit more about the patient engagement activities, just to note that we are continuing to work with our patient partners and obtaining feedback on things like the informed consent document. And so at this time, we continued to have some discussions on how we might be able to better support trial participants as the trial continued. And so something that came through from these discussions with our patient partners was that we might be able to develop some resources to help with the informed consent process. And so this actually led to the idea for a new project, which you're going to be hearing more about from Dr. Manoj Lalu this afternoon. And the goal of this project is to build a model for engaging patients in very early phase clinical trials. And so the idea for this program is to continue our patient partnerships throughout the CLICA-1 trial and kind of expand this model to additional clinical trials as well. And then the final new project that we'll also be hearing about this afternoon from Joshua Montroy builds on some of the work from the systematic review from GoCART. And so really the team was interested in taking a bit of a deeper dive into some of the data from the systematic review to get a better look at what factors or characteristics might be affecting the effectiveness of CAR T-cell therapy. So that's just a little bit of a summary on some of the projects that we'll be hearing more about this afternoon. And so at this point, I'm just going to hand things over to Terry so he can provide you his summary as well.
Okay, let's see. All righty. Yeah, good afternoon, everybody. I'd like to thank BioCanRx and partners for the opportunity to speak to you today. It's always great to have an opportunity for patients and family caregivers to share their experiences and thoughts, including sharing one's patient engagement experience. So a bit about me. My name is Terry Harish, and I'm a cancer survivor of about 10 years. Diagnosed with rare, aggressive, and unfortunately incurable form at this point of a non-Hodgkin's lymphoma. Went through an aggressive treatment regimen that was successful and achieved a stable remission, but of course not without some side effects that linger to this day. I'm a professional engineer by training and experience, and also an advocate for, I guess, positive patient outcomes in healthcare. And I also have a particular interest in patient engagement and support of clinical cancer research. So having an opportunity to collaborate as a patient partner with such a great group of presenters has been a real positive experience. And I'd like to think that we have learned from each other. Okay, so as you know, CAR T therapy holds some pretty great promise as seen in results from past clinical trials, and high response rates from I guess the high response rates and the effectiveness and the potential of CAR T as a cure for those blood cancer patients with limited therapy options makes this a compelling therapeutic choice. And today we will hear about a Canadian-based CAR T therapy research program that will eventually enable the point of care delivery of high quality and cost-effective CAR T therapeutic options to patients in need. My talk will also briefly look at my interpretation of the how and why of applying patient engagement to this research. And I'll also describe some key activities that I participated in as a patient partner, along with some personal perspectives. And My talk will parallel some of Madison's talk with the difference being I'll attempt to be even more plain language than Madison. So we'll see what happens. Next, whoops, where are we? Okay. So here's a slide that touches on the aim of our patient engagement activity. I was recruited from the Leukemia Lymphoma Society, where I do volunteer work, and was introduced to the research project by Dr. Manoj Lalu, principal investigator with the Ottawa Hospital Research Institute. And I was one of 2 patient partners recruited, my counterpart being none other than the one and only stuntman Stu Schwartz. Both of us were brought on board as research team members to help design an early phase clinical trial called the CAR program. Unfortunately, very few clinical research projects have reported in any significant detail on patient engagement methods and contributions. And one significant segment of this research was the use of patient engagement methodology, which was evaluated, reported, and published by the research team, including Stu and myself as co-authors. This is useful in that that this patient engagement approach can be used by others and adapted for the design of future CAR T clinical trials and potentially for other cancers as well. So we'll talk a little bit about the CAR program, getting better outcomes with the Chimeric Antigen Receptor Therapy. So the goal of the program is to build a better, I guess, a superior early phase clinical trial design for the CLIC-01 clinical trial that's currently underway, and using input from the participation of patients and other knowledge user groups, such as clinicians and policymakers. CAR program is about taking bench to bedside CAR T research to, I guess, a level that delivers superior patient outcomes. And this pilot project is going to create a template for design of future trials and therapy evaluations. So I'll talk a little bit about the Excelerator framework. Essentially, it's a method used by the research team, which defines 5 core sub-studies that are used to develop the CAR clinical design. And these sub-studies focus on improving where clinical trial designs have failed in the past. And this framework supports a collaborative team science approach that utilizes multiple user expertise outside the research team, including patient partners, health policy folks, regulatory folks, people that are involved in economics and clinical specialists, etc. Getting into the first sub-study of appraising preclinical and clinical available data, which is essentially a systematic review. And here we, patient partners provided input into to the research team on outcomes that we felt were important to patients. For example, quality of life factors. And as a side note, during the systematic review of, I think, about 60 trials that took place, was found that none had reported on these patient outcomes. We also worked with the team to develop a plain language summary of the results, and the review designed to be understood, of course, by a wider audience. And this summary and early identification of important patient outcomes was used to help guide and optimize the design of the CAR T trial as we were going on. So the second sub-study, implementation diagnostics, otherwise known as barriers and enablers to trial delivery. This included supporting the development and analysis of an interview to gain feedback from patients and caregivers living with blood cancer. My involvement was to review and help shape an interview guide to interview these folks and help to identify where interviewees could be sourced and to review these results with the research team. The patient partners also reviewed patient educational materials such as trials recruitment information sheet to ensure that it would provide clear information to patients considering early phase clinical trial participation. And in addition, we also helped create a letter of support of this research activity to the REB, or the Research Ethics Board, which appears to have had a successful conclusion. Okay, the next sub-study, early economic analysis, or the cost effectiveness assessment of CAR T therapy during early phase trial periods. Yeah, this sub-study contributes towards an assessment of cost effectiveness and I guess establishing a realistic commercial valuation of the CAR T therapy. And here I provided some insights on costs that patients and caregivers undergoing treatment are burdened with once they're outside the clinic. These costs included out-of-pocket costs during the treatment and recovery period, as well as other costs associated with impacts to lifestyle and personal productivity. And these things are not typically recognized in an early economic analysis. And also to help capture these stakeholder costs from a larger patient caregiver community, they also help develop and deliver 2 stakeholder engagement sessions that you'll hear more about from Mackenzie. Now, the next 2 sub-studies that I'm going to show, we didn't have any direct participation in other than attending meetings and gaining some knowledge on the topics. The first one being the testing of study feasibility or the retrospective cohort study. The second being the testing and validation of the therapy, the manufacturing, testing, logistics, quality control of the CAR T product manufacturing. But again, we attended meetings and started to learn, but we weren't directly involved. So I'll talk a little bit about the trial protocol and participant resource development. These are activities that my interpretation is that these are activities supporting and enhancing the Excelerator program. These things included some very interesting things to work on. One was a visual informed consent that was created along with a non-technical summary of the trial program to help patients clearly understand from day 0 to the end of their treatment period exactly what takes place during the clinical trial, to enable them to better evaluate the risk and reward associated with signing a informed consent, which is a very stressful and complicated process for the typical patient. Some of the other involvement activities that weren't really part of the sub-study boxes, we input on recruitment approaches for soliciting patient partners in the future, developed a terms of reference document that really captures all the roles and responsibilities and timing and who does what associated with the GO-CAR program that can be modified and built on for future studies. We had the ability to present our cancer journey to the research team, which I think is always good and helps excuse me, ground them a bit in terms of understanding again what they're there for. Participated in some great onboard training and involved in a grant application that helped support the GO-CAR program. So that's some great stuff. Now, just to really, really briefly talk about some future projects. You already heard about the Marvel program that Madison talked about. I won't get into it in too much detail. We'll hear a lot more about it in subsequent presentations, but there will be patient engagement activity associated with these studies, which is great. And another one, the identifying of effect modifiers of CAR T-cell therapeutic efficiency. Again, another future study that will hopefully require some deep patient engagement to support as well. So, what are the positive study outcomes associated to the patient community? This results in patient engagement results in more relevant research results, and the knowledge and lived experience of cancer patients is just it's not a checkbox. It's really valued as input. In patient engagement in this study is resulting in issues that are important to patients being built into the design of the clinical trial. More realistic economic modeling with patient input is achieved, which helps guide the trial design and results in realistic commercial pricing of the therapy. Improved clinical trial patient recruitment, retention through the understanding of barriers and enablers. Helps to improve the transfer of study results to other user groups that participated in the trial study, for example, clinicians, and a more effective therapy design by knowledge users, and including the patient groups. So, what does this do? This information generated, translated, and transferred from the study will allow patients to make better informed decisions on CAR T as a therapy choice. And, we patients are ultimate end users and customers of the knowledge and products and services generated. Patient engagement helps to direct research to areas of priority to the patient. And just some final perspectives concerning my engagement. I felt that it was a very positive experience. Excuse me. I felt like a true partner, and had a great feeling of accomplishment for being able to provide this type of input. Some of the great things I felt were the induction into the studies and project and the associated training, terms of reference development, the showing of mutual respect between all parties. Everybody had their say, including the patients. The research team was flexible and accommodating in terms of our time. A couple of things to work on. Recruitment of patient partners could be maybe a little bit more effective. Diversity issues in the patient partner group, and of course, never enough training. And compensation, that's interesting, hot button issue, but certainly well worth looking at. So if a patient partner opportunity presents itself, go for it. And hey, inaction never leads to change or progress. So thank you very much.
Terry, thank you very much for that. I think that quote was a perfect segue. So I think at this point, I'd like to thank both you and Madison for the plain language summary and for your perspective, Terry. Just a reminder to all participants, we are actually taking questions at the end. If you do have a question though at this point, I would recommend that you put it in the chat, or rather, excuse me, in the Q&A box, and that way we can get to it. So we're moving on to the talks by the other speakers, and we'll actually come back to Madison and Terry at 3 o'clock. So from 3 o'clock to 3:15, you'll be presenting any responses to Q&A that are directed at you. So again, Terry, I'd like to thank you. I'm going to share my screen now, just ever so briefly, to introduce the remaining talks. So let's see if this works. Yes, I think it did. So that's perfect. I just want to make sure I have everything right. Okay, so welcome for those of you who have just joined us. You are attending the second session of the BioCanRx Summit Speaker Series that was developed in lieu of our in-person Summit for Cancer Immunotherapy conference. And we thought that this was a really good way to engage the community and to have an opportunity for our network investigators to present their data and have our stakeholders attend and engage as much as possible in this virtual setting. So at this point, I would also like to mention that we will be putting the agenda and other technical notes in the chat box. So I'd refer you to there for more information. And at this point, I'd also like to mention that the session will be recorded. For any questions that you have for the speakers, please do put them in the Q&A box. And if you are experiencing any sort of technical issues, please put those in the chat box. So to keep it short, I'd like to hand it over to Dr. Dean Fergusson, who is a senior scientist and program director of the clinical epidemiology program at the Ottawa Hospital Research Institute and the Ottawa Hospital. So Dean, with that, I'll let you share your screen.
Great. Thank you, Megan, and Madison is going to run the slides for me. So I'd like to thank everybody for tuning in. And thank BioCanRx for sponsoring such a session. As Megan said, my name is Dean Fergusson, and I'm a senior scientist at the Ottawa Hospital. And with my colleague, Dr. Lalu, we put together a program that tries to enhance clinical translation, or bench to bedside, as they say. And both Terry and Madison gave a great overview of our Excelerator approach that we use for the CAR T program, called GoCART, and the trial being called the CLIC-1 trial. And thank them both for that overview of sort of the components that went into it. And I thought I'd just, next slide there, Madison, I thought I'd just take a bit higher view, sort of at 50,000 feet, if you will, and talk about two important aspects of our approach in building clinical trial protocols. The first is assembling the team. So, identifying the necessary expertise and bringing that team together, breaking down silos within disciplines and across disciplines is the aim there. And aside from it being a necessity. And two, once you have the team assembled, is together building the research agenda that will build a robust protocol. So in the past, thanks, Madison. So the team science, in the past, it was usually, a couple scientists coming together with the clinical side, clinicians, and manufacturing, and they would sort of hammer out a trial protocol. And we think a much more robust approach to ensure better translation and better uptake of the trial and having more informative results, regardless of the results, to be frank, is to involve different disciplines. And so from stats and design to knowledge translation to ethics to patient involvement, health economics, a ton of expertise is required to building a robust trial protocol. And so that's what the GoCART funded by BioCan helped us develop the sort of conceptual model of the Excelerator conceptual model. And behind the scenes, what was at that is the team science approach. Next slide. And so once you've built that team, then you build the research agenda. It's not just a single study. It's not just that design of the protocol. There's a bunch of other work that needs to go in, and that's what you've already heard about, and you'll be hearing about some in more depth, over the next hour or so. And So, doing systematic reviews of the clinical evidence, the animal evidence, the manufacturing evidence. And so you have up-to-date evidence that can inform the trial on eligibility criteria, on the outcomes, on the manufacturing technique, and different aspects there. Patient involvement activities that Terry has been a huge help on with the GoCART. Observational studies to justify your eligibility criteria and sort of trial run your eligibility criteria, so you don't underestimate or overestimate, I should say, the time that the trial will take in terms of patient enrollment. And you'll hear a bit more from Gisell today on knowledge translation, all the interviews and potential surveys and so forth to help inform the trial and sort of take the pulse of patients and their family members and clinicians. So putting all that together in terms of building the team and building the research agenda is sort of the higher picture view that went into the GoCART program. And like I say, you'll be hearing about pieces of those, including next steps and to sort of further refine the approach and make it more generalizable to all researchers. And I think that's what's an important message as well. It's, again, not just that phase 1 trial. It's all the scholarship that each of these individual studies supporting the trial are worth in terms of helping other researchers in developing their programs. And importantly, today you'll also hear from our trainees and our HUP, and it's really exciting that previous trainees are now within our team, such as Madison and Josh, and I think that's been one of the most exciting components of this, that when you build robust research agendas, not just the trial, but robust research agendas, that it offers more opportunity for different disciplines to come in and help with the translation process. So with that, I'm going to turn it over to Dr. Thavorn, and Kednapa is a senior scientist and a health economist at the Ottawa Hospital Research Institute and professor with the School of Epi and Public Health at the University of Ottawa. And she's going to talk about some of the health economic aspects that went into the CLIC-1 trial design. Kednapa.
Thank you so much, Dean, for a really nice introduction. Today, I'm going to talk about early economic evaluation, and our HQP, Mackenzie Wilson, will talk about stakeholder engagement. Make sure that everyone on the same page. I just want to remind everyone that this particular project, we focus on health technology assessment, or an HTA, but the HTA that we're focusing on is actually happening in an early stage of the development, or an early phase trial. But we're focusing on the same component as the mainstream HTA, where it's looking at whether or not the intervention safe, whether it's work, or whether or not it's actually offer good value for money. The target audience of an early HTA is actually a manufacturer, product developer, or investor. It can help inform the design and management of a technology, and also regulation of reimbursement strategy. For the early economic evaluation and also early HTA, it's typically based on evidence from early bench and animal testing, early clinical experience, or it could be even based on previous generation of the technology. For this particular presentation, I would like to focus on one aspect on early HTA called early economic evaluation, different from the conventional and also traditional economic evaluation, where it typically happens at the end stage where the new product is about to be available in the market and to be reimbursed by healthcare payer. Early economic evaluations actually help to set a benchmark for the minimum performance that is required for the new technology to become cost-effective compared with current practice. At the same time, it also can provide insight into the maximum additional cost at which the intended use of the new technology is deemed cost-effective. In other words, we actually trying to do a backward calculation to under the assumption that if the technology is work as planned, what would be the maximum price that it can be set to ensure that this particular technology is still considered cost effective. This particular type of analysis can be also help to inform whether or not additional research is required, and with the limited budget in research funding, how we should prioritize research agenda. For this particular exercise, we actually use economic evaluation to actually looking at adult patients with advanced leukemia, specifically relapsed refractory B-cell ALL. We compare CAR T therapy to currently available treatment, including blinatumomab, inotuzumab, bone marrow transplant, and salvage chemotherapy. All these links are obtained from a consultation by critical experts who are part of our team. The outcome that we're looking at is actually maximum reimbursable price, in which the maximum cost at which the CAR T therapy can still consider cost effective. And at the same time, we're also looking at whether or not are there any value of conducting future research in the trial. And our analysis is conducted based on from the perspective of Canada healthcare system. This particular slide just showing the maximum reimbursement price of CAR T-cell therapy. As you can see, it's actually the price vary according to how much money policymaker willing to pay in order to improve one unit of outcome. If you're looking at, like, for example, when comparing with blinatumomab, the price of the CAR T therapy that can set ranging from $132,000 to about $148,000. And it's also vary according to the type of comparator being compared with CAR T therapy. This particular slide looks scary, but it's pretty, it conveys a very simple message. On the y-axis showing the monetary value of future research, the x-axis showing the price of potential price of CAR T therapy. As you can see from this particular slide, the future research will actually be worthwhile if we're focusing on the impact of CAR T therapy on relapse rate. And the higher the price of CAR T therapy, this particular outcome is even more valuable. So in conclusion, what we have learned from our exercise is that the maximum reimbursement price is actually depend on a number of factors, including what we should compare in terms of the currently available treatment, how much money policymaker willing to pay in order to improve patient outcome, and also a particular line of therapy that CAR T will be considered in the clinical practice. And given the limited budget in research funding, future trials should focus on the effect of CAR T therapy on relapse rate, and at the same time also overall survival as well. It should be noted that our analysis is actually based on an early evidence, and at the same time with a limited number of stakeholders. Mackenzie Wilson, our HQP, actually gonna walk us through in terms of how we're trying to improve and validate our result and the economic model that we develop. And now I'm going to turn on Mackenzie. It's your turn.
Great, thank you, Kednapa. Alrighty, so building on what Kednapa has discussed, we are engaging various stakeholders in the project to provide their input and their perspectives on the economic evaluation. So we're involving patients, clinicians, and researchers, as well as healthcare payers and policymakers. So traditionally, stakeholder engagement in economic evaluations has included clinicians, policymakers, and reimbursement decision makers. However, other stakeholders, such as patients and their caregivers, have insight into factors that can improve the validity of an economic evaluation. So for example, oncologists and hematologists are familiar with the therapeutic options for treating blood cancer and the sequences of therapy and can inform the scope and inputs of the economic model with their clinical experience. And on the other hand, patients and their support network are familiar with the resources that they require for treatment. And so they're best placed for describing outcomes beyond recurrence and mortality, such as the side effects and quality of life factors, for example. So without input from all key stakeholders, an economic evaluation model may use insufficient or even inappropriate assumptions. So we are involving healthcare professionals, policymakers, and patients and their support networks to inform the conduct of an early economic evaluation. And so by involving these stakeholders, we hope to deliver on a few study objectives. So the first one being the generation of stakeholder-specific knowledge to inform model development based on the experiences of key stakeholders. So we are holding discussions with patients and their caregivers to understand the costs that are important to them when receiving treatment that might not be otherwise considered, such as those associated with transport, accommodation, or the emotional costs, for example, as well as the important benefits of treatment, such as the limited side effects or ease of treatment delivery and etc. And second, we hope to verify the comparators or the treatment strategies being considered. So we'll do this by involving clinicians, researchers, and healthcare payers and policymakers. So we'll rely on the expertise of these individuals to provide insight into the course of treatment and the associated clinical assumptions to make sure that the model reflects clinical practice and is also useful for healthcare decision makers. And finally, we hope that engaging these groups, that we will collect and seek their feedback to identify the steps and ways that they can, but also want to be engaged in this process. And so using this feedback, we will develop a framework for a specific method of stakeholder engagement in future similar economic evaluation studies. So far, we've conducted discussions with patients and their caregivers, and they have provided their perspectives on the costs and benefits that should be considered in the conduct of the early economic evaluation. So for example, they've highlighted the financial costs of treatment and side effects, such as the financial costs associated with transport, accommodation, food, and the out-of-pocket drug costs, as well as the costs associated with lost income, missed promotions, and interruptions to education. They've also highlighted the importance of the physical, social, and emotional costs of treatment. And while it is difficult and in some instances impossible to put a monetary cost on some of these considerations, it is nonetheless important to recognize these and highlights potential avenues for future research. So similar to other fields of research where challenges exist in translating research into practice, the limited use of economic evaluations in the allocation of healthcare resources has also been recognized. So in involving these stakeholders, this represents an avenue to potentially enhance health economic evaluations and to support their use to inform treatment research and development and facilitate future market access. And so to continue to move this work forward, we are aiming to develop methods specifically tailored to engage healthcare professionals, policymakers, and payers. And then we hope to combine all of our findings to inform that tailored framework for stakeholder engagement in future similar studies. So thank you for your attention, and I am going to relinquish control now and hand it over to my friend and colleague, Gisell.
Okay, great. Hi, everyone. Can everyone hear me well? Great. So thank you for joining us today. I'll be discussing highlights from 3 sub-studies we conducted as part of the Excelerator model to support the CLIC-01 CAR T-cell therapy trial. Sorry, Madison. Oh, okay, there we go. Working from an implementation science perspective, we know that factors that encourage and prevent our actions are not always obvious to us. So if we can identify what those factors are that encourage or prevent the likelihood of us engaging in a specific behavior, we can then develop strategies to support those behaviors. So prior to the launch of the CLIC-01 trial, we conducted 3 sub-studies in order to identify what are the barriers and enablers to hematologist screening for the trial, and what are the barriers and enablers to participating in the trial from the perspective of people living with blood cancers. Oh, sorry, that just popped out. One moment, please. There we go. So to guide our study design and analysis, we use the validated version of the theoretical domains framework, which synthesizes constructs from 33 behavior change theories into 14 domains or broad topic areas that help us understand what factors may help or hinder those specific behaviors we're interested in supporting. the three sub-studies that we conducted were an interview study with hematologists, an interview study with people living with blood cancers, and a national patient survey. So first, let's turn to the hematologist interview study. We completed 15 interviews with hematologists across Canada. We conducted a directed content analysis, identified relevant domains, and generated global themes across domains. So what did we find? Our first theme, Cautious Hope, captured an underlying tension where most hematologists reported that they intended to screen for the CAR T-cell therapy trial because they believed the trial would benefit patients. However, at the same time, they also voiced concerns regarding the potential for adverse events, as well as concerns regarding the uncertainties associated with a novel therapy. Understandably, they expressed wanting to know more about toxicity management and response rates. Many were also concerned about the feasibility of the CAR T-cell therapy trial because they believed that finding eligible trial participants would be difficult. Many therefore suggested that eligibility criteria must be broad for the trial to be successful. Encouragingly, every hematologist in this study believed that screening was part of their role or job description. However, they described that screening and recruiting practices at their particular sites tended to vary quite a bit. And at times this variability improved screening rigor, but at other times it potentially might lead to missing trial candidates. Hematologists also described working in challenging contexts. So for example, they noted that trial sites would need resources to complete diagnostic tests, fund statuses with screening, and to provide financial supports for participating patients. They also noted that trial protocols that were too labor intensive would likely be a barrier. And so they suggested that amidst busy clinics, sometimes they may forget to actually screen for the trial, and so reminders would be useful. Okay, let's turn to the patient interview study. In total, we completed 13 interviews with people living with blood cancers. And like in the previous one, we conducted a content analysis and generated global themes. Our first theme here was similar to hematologists, in that people living with blood cancers believed they would benefit from the CAR T-cell therapy trial, even though they knew that the intent was to assess safety and feasibility rather than efficacy. They were also concerned regarding the risk of adverse events and were uncomfortable with the uncertainty of outcomes. Regardless, they suggested they would be motivated to participate, especially based on altruistic considerations like benefiting future generations or advancing science, but as well, we're interested in the potential for personal benefit, as I mentioned. To better navigate uncertainty, participants indicated they would appreciate receiving accessible information about treatment safety, response rates, and trial logistics, especially in a way that they could share with their family members and loved ones. Our second theme speaks to who participants viewed as influential. And so even though people like family members and other clinical staff were mentioned, most participants emphasized the importance of hearing about the trial directly from their hematologist or oncologist. Participants also varied in their expressed intent to participate in the hypothetical trial, or hypothetically at the time. Many suggested they would be more likely to participate if they would benefit from it, if they had no other treatment options, if their doctor asked them to do so, or if the trial was offered at a later stage once safety had been established. And then last, similar to hematologists, interview participants emphasized the need for resources. They indicated that having expenses paid for, like parking, accommodations, caregiver costs, would make participating in the trial easier. However, not all participants suggested they would need supports. So if they lived close to the trial site, for example, they wouldn't need parking house coverage. Others stressed the importance of having access to social supports like health advocates, social workers, and counseling services. Okay, so to complement our qualitative research findings, we developed a national patient survey based on the theoretical domains framework. Our goal with this survey was to better understand the views of people living with blood cancer regarding their thoughts on participating in a hypothetical CAR T-cell therapy trial, as well as to assess what factors or theoretical domains were associated with an intent to take part in the trial. So in total, we received 115 complete survey responses. And now looking at what we asked our respondents about their past experiences with clinical trials as well as information needs. Notably, 80% had never taken part in a clinical trial before. And similar to our interview study, most participants wanted to know more about side effects, response rates, trial logistics, as well as general information on how clinical trials work. They also, they also indicated that their preferred way to receive information would be by hearing it from their doctor. Okay, so looking at the correlations that we ran, and this is looking to see what associations there are between intent and the theoretical domains. We found that social role and identity, beliefs about consequences, emotions, goals, and social support were all correlated with an intent to participate in a CAR T trial. We then ran what's called a multiple regression analysis to see which theoretical domains were able to predict intention. Here we see that social role and identity, beliefs about consequences, and goals were all positively associated with an intent to participate, and that the emotion domain was negatively associated with intention. Overall, these 4 factors accounted for 81% of the variance in intention, which is a fair amount. Social support, however, which we saw was correlated with intention in the previous slide, was not a significant predictor of intention. So looking at the items on which these 4 domains were based, we see that the decision to participate in a CAR T trial is indeed multifaceted and complex. And so importantly, it becomes clear that decisions are based on more than just information. This tells us that with a better understanding of how these factors contribute to patient decision-making processes, we can then develop tools and strategies to better support people who are considering taking part in novel trials like the CAR T-cell therapy trial. So taking all this together, the results from these 3 sub-studies have important implications for the conduct of ongoing and future CAR T-cell therapy trials. For one, both patients and hematologists would need accessible and up-to-date evidence regarding safety, efficacy, and toxicity management. The role of hematologists and oncologists was also highlighted as critical, given that patients clearly stated a preference for receiving information directly from them. Hematologists and patients also emphasized the need to provide a variety of resources to support trial participation, particularly in the interview studies. And finally, the results of the patient survey highlight that meeting patient information needs may not be enough to support decisions, and instead, more work is needed to understand the relationship between intention and the 4 predictor domains we found were associated with intent, social role and identity, beliefs about consequences, emotion, and goals. So, and with that, I just want to say thank you to everyone who contributed to these studies. They would not have been possible without you. And now I'm happy to turn it over to Dr. Manoj Lalu, an associate scientist at the Ottawa Hospital Research Institute and an assistant professor in the Department of Anesthesiology at the University of Ottawa. Thank you.
I was warned it would do that. I almost started rearranging the slides. So yeah, thanks, Giselle, for the introduction. I'm an anesthesiologist at the Ottawa Hospital and associate scientist. As she mentioned, I'm also in the OR today, but someone else is taking over the case, obviously, at this point. So, just over the next 10 minutes or so, I'll give you a quick rundown on what we're planning for the next 3 years or so with our MARVEL program. And Madison has already mentioned this in some of her introductory slides as to what we're planning, and I'll just give you a few more details, basically. So going back to the Excelerator program, which you heard all about, and you heard different components of the Excelerator program already, and specifically the Excelerator that we had put together for the CLIC-1 trial we had called GoCART. That's why that GoCART term is on the top here. Basically, throughout all of these aspects of the Excelerator, we had patient engagement, and Terry has provide a little bit of an overview of how that actually occurred within these component projects. Someone's already actually asked about our write-up of this, and I've provided a link within the answer box there. So if you're interested, you can take a look at that. But again, throughout all of this, we had patient engagement where we thought we could incorporate it. And then for the couple of boxes that Terry had already pointed out, well, there was nothing there at that time. We're already starting to try to figure out, how we might actually have patient engagement within these other aspects as well too. And this is just a screenshot of that paper where we had basically summarized our patient engagement program within GoCART, and specifically how Terry and how Stu were involved as patient partners within it. So at the end of the GoCART program, we actually got another small grant to start to think about how we might continue to have patient engagement within the CLIC-1 trial as it was initiated at that point. So everything GoCART related really was a lead up to initiating the CLIC-1 trial, and how we might optimize that trial to make sure it becomes a success. So at the point where the trial became initiated, we got the small grant from the Ontario Spore Support Unit, where we got to get together with Terry and Stephanie Dupont, who's another patient partner. Some of you might have heard her speak previously, as she did speak at one of the BioCanRx summits a couple of years ago. She actually had received CAR T-cell therapy in the States, and she spoke about her whole experience there. So obviously very well-informed patient perspective as we start to think about the CLIC-1 trial and how we might have patients engaged as partners throughout that trial. And this is just a screenshot, a photograph of one of our face-to-face meetings where Dr. Kekre, who some of you heard her speak a couple of weeks ago, basically she's one of the lead investigators of the CLIC-1 trial. And then a few of the other folks that you've already met online here through these presentations. And from this, we basically started to think about, okay, what can we practically do? And these are three things that we've came out of that award and that we've actually implemented within the CLIC-1 trial. So the first is a non-technical summary of the informed consent document. For those of you that may have been involved as a participant in the trial, you probably recognize that the consent documents are usually fairly large, tens of pages often. And to basically demystify some of that, we put together, along with Terry and along with our other patient partners, a one-page summary of what this informed consent document was actually all about. As well, along with that, we had a visual informed consent document. So I'll show you an example of that in the next slide, and a glossary, again, just to demystify a lot of the jargon that is usually thrown around in these sorts of clinical trials. So this is a part of the visual informed consent document where we basically show, and this is given to the patients themselves along with the usual informed consent document, where they have day 0, where they're actually going to have the CAR T-cell therapy infusion, what happens 14 days before, what happens the months leading out from that infusion. And here's just a visual representation. Okay, they're going to have a half a day here, a half a day here at the hospital. They have a hospital with a moon here, so that represents they're going to be there overnight, potentially for a few days as well too. So it's a very quick overview of what they might anticipate if they are going to participate in this trial. So again, this is part of the informed consent document process, and this is all put together in collaboration with our patient partners. So this is, again, just trial protocol and participant resource development. That's what that was all about there. In addition, we said, hey, well, maybe we could do some of what we've already done with GoCART, plus some of this ongoing engagement of patients as partners in the trial itself. Maybe this is something we could roll out to other BioCanRx funded trials. And that's exactly what the MARVEL program is all about. So MARVEL stands for Making Patient Partnerships a Reality in Very Early Phase Clinical Trials. So the objectives of this particular program are to develop a platform for meaningful patient engagement in early phase clinical trials. Two specific things that we thought we could do was build patient panels, and those panels would be the patient partners for up to 4 BioCanRx funded clinical trial programs. And as well, we are going to seek to describe our methods, processes, activities, and lessons learned in a series of manuscripts and policy briefs, et cetera, that we hope to share with all of you in the next few years. Specific key deliverables within the MARVEL program. First of all, whenever we have patient partners involved in either what you saw for the GoCart program or other patient engagement projects that we have on the go, we always put together a terms of reference with our patient partners to make sure we're all on the same page as to what we expect, what they expect, et cetera, and what roles might be as we move forward. We're also going to be co-developing patient and caregiver documents, toolkits, and resources. So an example of that would be the visual informed consent document that I've shown a previous slide on. We want to coordinate with existing patient organizations to set up trial-specific peer support groups. So that means that patients who are actually be participants within the trial, they might have someone to reach out to for peer support as well too. So this will be something that we'll be looking to set up over the next couple of years. Finally, evaluation of strategies, which is an interesting area. Within patient engagement as a science, there's an increasing number of people doing this within their research programs. Top tier journals like the British Medical Journal, for instance, they now have required sections in every single article they publish as to how the researchers actually engage patients as partners. However, I think one of the knowledge gaps that we've identified and other groups have identified is actual evaluations of the patient engagement and how to, again, further demonstrate value for engaging patients as partners. And then finally, dissemination of findings. So within BioCanRx, as well as other networks, we'll be looking to disseminate some of our findings and hopefully encourage other researchers to engage patients as partners within their research programs. Within MARVEL, we've already identified 4 trials, and I say that with some hesitancy because, as I'll show you, we're actually now going to have a whole spectrum of research from very late preclinical research to actual trials that we'll be engaging patient partners in. So, for instance, we have one program which is an enabling study. So this is, again, very late laboratory research, moving into the point where they could start to consider clinical trials around this product. And this is in collaboration with the NRC and a few of the other BioCanRx, NRC's National Research Council, and a few other BioCanRx investigators, where they are looking to develop CAR T-cell therapy for B-cell leukemia. We also are collaborating with the THINK group, and this is another funded trial. And they are going to be moving in, probably in about 2 years' time, into a phase 1/2 trial. At this point, they're doing a lot of things that we did for the GoCart program leading up to the CLIC-1 trial. So they're in a similar state, so where we could actually engage them in a lot of these early studies that will hopefully help them optimize their clinical trial protocol. And that's led by Dr. Michelle Duval, Duval's group. And then the CLIC-1 trial, I'll just move to number 4 here for a moment. First, you've heard all about it, and we're going to try to continue patient engagement within that clinical trial and hopefully provide some more value add in terms of patient engagement within that trial and help those participants within the trial themselves be heard hopefully a bit more as well. Finally, I will also mention a trial that's being led by Simone Turcotte's group. They actually have another research group that's going to be quite involved in their patient engagement program. However, we will be collaborating with them as well. And they're looking at another cell therapy for refractory melanoma. So what's really nice and what's going to happen over the next 2 to 3 years with this funded program that BioCanRx is supporting is that we're going to really have patient engagement over a very wide spectrum of clinical trials and clinical related studies as well too. So last slide here, basically, in terms of what we're thinking about. I've already talked about terms of reference, informed consent documents. We'll have inputs on the protocol as they're being developed. So we'll have patient partners, really help us figure out what the barriers and enablers might be, what the costs might be, what are some of the best ways to recruit patients as participants into these trials. What might patients tolerate or not tolerate in terms of schedules of assessments afterwards. and also trying to help determine other patient important outcomes and identify those and hopefully incorporate them again within the protocols of those trials. We're hoping also to develop training and toolkits for researchers and patient partners. So how do we actually do patient engagement? How can we make it better? And these toolkits will provide some of a framework for this. And then finally, I already talked about patient or peer support panels, leaflets or other materials for further education for trial participants within these trials as well too. So lots of exciting ideas as we start to move forward with all of this. This is my last slide, just to acknowledge a very large number of people who have been involved in all these studies over the last 3 years now, and will be involved over the next 3 years as well too, as we start to roll out Marvel. Thank you.
Oh, great. So hi, everybody. My name is Josh Montroy. I'm a clinical research associate with the Blueprint Group. And today I'm going to briefly talk to you about the work we're doing to build off our original systematic review, a project entitled Identifying Effect Modifiers of CAR T-Cell Therapeutic Efficacy. So we've seen this figure quite a few times over the last hour, but my talk is primarily going to be focused on the orange piece here at the top, which is the appraisal of preclinical and/or clinical data. So in our previous appraisal of data of CAR T-cell therapy, we focused primarily on clinical trials, and we identified 42 studies of CAR T-cell therapy in hematologic malignancy patients. Across all studies, we identified about 54% of patients experienced a complete response. And when we looked at safety outcomes, we found 55% of patients had cytokine release syndrome, and a little over a third experienced neurotoxicity. So this appraisal of evidence led to a publication of a systematic review about a year and a half ago in Transfusion Medicine Reviews. But even with the completion of this project, there was a lot of great things that came out of it. A lot of it went into informing pieces of the clinical trial, as you've heard throughout the last hour. But there was still a lot of questions that we had going into the project, which we couldn't answer with the approach that we originally took. So, for example, what was leading to the incredible variability in responses that we were seeing in these clinical trials? So in our sample of 42 trials, we had some trials reporting 6% of patients experiencing complete response, and other trials reporting 93% of patients experiencing complete response. So what was the reason for this? And we couldn't tease this out given our approach. Second, we wanted to know if there was a correlation between the efficacy, so patients experiencing complete response, and the cytokine storm, so safety effects such as cytokine release syndrome. Again, we couldn't tease this out from our data given the approach that we took. And lastly, we wanted to know if we could identify patients or interventional traits which may lead to better or worse therapeutic efficacy or increased or decreased safety concerns. And again, with the approach that we took for this systematic review, we were unable to answer this question. So traditionally, when you see a systematic review, and what we did for our original appraisal was what's known as an aggregate data meta-analysis. So we have this population here on the left, which is we'll call it our trial population. So this is 30, 40 patients who were included in the trial of CAR T-cell therapy. And what we do is we identify all these studies and we extract the data. So first we extract some participant level or some population level data about the patients themselves. So we extract median age of the trial cohort is 50. And the median dose given to the trial cohort was 2 × 10⁶ per kilogram. And then we extract our outcome data. So for this whole population, there was a complete response rate of 58% and a cytokine release syndrome rate of 42%. So the reason that we were unable to answer some of the questions I showed on the previous slide is because of the incredible heterogeneity and variability in these characteristics, not just across the 42 trials, but within each individual trial themselves. So for example, we had a median age of 50 in this trial, but the range was 19 to 72. And the dose ranged from 1 × 10⁶ to 3 × 10⁶. So you can see why this may produce variable safety and efficacy outcomes. So again, we saw 58% complete response rate in this population, but we don't know who exactly is responding. Is it the young patients, old patients, men, women, patients with higher dose, lower dose? Because we're extracting data on a population level, we don't actually know. So the way we can answer this is by taking a bit of a deeper dive into the data and performing what's known as an individual patient data meta-analysis. So this goes one step further from what we originally did, or I guess a couple steps further, and takes this same population that we had on the previous slide and essentially, breaks it down further into and extracts data at the individual patient level rather than the whole population. So rather than the median age of 50, we have patient data for all these patients. So example, patient A, he's 79 years old. This is the dose he got, and these are his outcomes. So he achieved a complete response and he had no safety concerns. And then we have patient B, significantly younger. This is the dose he or she got. He did not achieve complete response and he had safety concerns, and so on. So we do this for every patient included in the trial. So as I mentioned a little bit on the last slide, an individual patient data meta-analysis is an analysis of raw individual level data rather than the aggregate level sort of population level data, which is typically used for traditional meta-analyses. So there are several well-documented advantages to this approach, but the one that primarily interests us and the reason we chose to take this approach is it gives you the ability to model individual level interactions, i.e. the association between a patient level characteristic such as age and an efficacy outcome such as response directly within the studies themselves. So the downside is it can be very time consuming and labor intensive. As you can imagine, if we have 42 studies and 30, 40 patients per study, and we need to extract data for every single patient, it can be very time consuming. And additionally, you need to have access to the individual patient level data, which especially with some of the larger industry funded trials isn't always possible. So with that in mind, the objective here is to perform an individual patient data meta-analysis exploring potential effect modifiers of CAR T-cell therapeutic efficacy. So additionally, we're going to update the previous systematic review, which I talked about on the first slide, to capture the growing evidence of CAR T-cell therapy and provide a contemporary up-to-date estimate of safety and efficacy. So very, very briefly, this is our PCOS criteria. So a PCOS criteria is sort of the backbone of a systematic review, and it details which studies are going to be eligible for inclusion in your review. And PCOS stands for Population Intervention Comparator Outcome and Study Design. So in our review, we're going to be including patients with hematologic malignancies who received CAR T-cell therapy. The studies do not need to have a comparator arm. In fact, as we learned in the first review, the majority of these studies are simply single arm trials. Our primary outcome is going to be complete response, and our secondary outcomes are safety, such as cytokine release syndrome and neurotoxicity, which I mentioned previously. And we're only including interventional trials. So we're not including anything, like a retrospective study, no case reports, anything like that. So we're going to go about this by performing a systematic review using gold standard systematic review methodology. I won't get into the nitty gritty, but very, very briefly, we have an information specialist search the sort of standard scientific databases such as Medline and Embase to identify abstracts which are potentially relevant to our study question. These abstracts are then screened in duplicate by two people to identify potentially relevant full text articles. This process is then done again on the full text articles, again in duplicate, to identify the studies which will end up being included in our systematic review. So this is a list of our covariates of interest right now. So these are the variables which we are going to examine to see if they have an effect on how effective and safe CAR T-cell therapy is in patients. So on the left, we have more patient-related factors such as age, sex, cancer type, the previous lines of treatment the patient received, their duration of follow-up, the duration of the response if they achieved one, and the type of lymphodepletion which was used. On the right, we have more intervention-related characteristics such as dose, the CAR T formulation, and the co-stimulatory domain used. So very, very briefly, for the analysis itself, we're going to choose a two-stage individual patient data meta-analysis approach, where in the first stage, interactions are modeled within a single trial itself, and associations are determined and effect estimates are obtained. And then those individual effect estimates from a single trial are taken together with all the other trials, and a more traditional aggregate data meta-analysis is performed. And we will be using meta-regression techniques, which are very similar to normal regression, to determine the association between the covariates on the last slide and our outcomes such as efficacy, complete response, and safety such as cytokine release syndrome. So right now, we've started this project. It's going to take us about a year. We're in the process of identifying the new studies for inclusion. So in our previous systematic review, the search stopped at about November 2017, so 3 years ago. And we've seen quite an explosion of CAR T-cell clinical trials in the last 3 years. So we're in the process of identifying every new trial that's been published in the last 3 years. And we have about 3,000 abstracts to go through. So we have a couple hematology fellows on board who are helping us screen through these trials currently. And that's it for me. Now I'm going to pass it off to Dr. Justin Presseau, who's going to lead us through the Q&A.
Great, thanks, Josh. I'm Justin Presseau, scientist at the Ottawa Hospital Research Institute. And we've got a few minutes for questions. And I'd encourage you to use the Q&A button at the bottom of your screen if you have a question and haven't posed it yet. And while you're doing that, there's a couple of questions that have already been answered, but I might, for the benefit of those who haven't been looking through the Q&A so far, I might just quickly touch on those. So Doug Buchanan asked, what's your best guess as to when this therapy might be standard of care? And Manoj provided some response to that it's indeed already approved for certain patients, and there's a good article on healthydebate.ca on the topic, and Manoj provided a link to that for anyone who's interested. Another question that came through is a question around whether patients, Sandra Donaldson asked whether patients have a say in what outcome measures in the trial are most important to them, and the use of patient-reported outcome measures. Manoj, did you want to field that? You may have been, oh, there you go.
Mute. So in terms of patient-reported outcomes, so what was really interesting from our last systematic review, which Josh just touched on, again, we only went to 2017, the end of 2017. So basically all the trials that we found published up to that time, none had actually reported on quality of life. Or any measures of quality of life. So that was an issue that Terry and Stu had brought up as something that was, obviously very important to them to understand, or what they felt, patients in that circumstance would want to know about, prior to agreeing to undergo CAR T-cell therapy, what their quality of life might be, 3, 6, 12 months, et cetera. After CAR T-cell therapy. So because of that, we actually incorporated quality of life as one of the outcome measures. It's part of a larger group of measures that we're taking through some questionnaires that the patients will be, the patients who are participants within the CAR T-cell therapy trial will be completing.
Great, thanks. There's a quick question about a link that I think Josh can address. But maybe while we're still waiting for more questions, and we've got a minute or so left, maybe taking it right back to Dean at the start around the idea of a team science approach and bringing this sort of program of work together. And I wondered if you had any advice for any PIs that might be on the call that might be considering taking on this broader program of work within their area. Where should they start?
Yeah, I think it's reaching out locally within your own institutions for, starting with sort of methodologists, and more institutions are having, special offices and frameworks and resources for patient involvement in research. So that would be a good start. And, usually a methodologist opens up to the other methodology. So a statistician or a trialist, they'll know health economists and those in the knowledge synthesis and knowledge translational world. And You know, we've certainly found that it works. What we really didn't talk about is that the efficiencies gained with all the upfront work in having trial protocols that are actually workable, that get through REB a lot sooner, get through regulatory approval a lot sooner. We've certainly demonstrated that, not just with GoCART, but in other areas. Yeah, and you know, tapping into the university level, you know, schools of epidemiology or health research methods is another opportunity as well.
Great, thanks. So we're running a bit short on time. We've got but one question, maybe a quick one from Sandra Donaldson again. For randomized trials, will the stats be stratified by center due to variability of the therapy, or has it become more standardized?
Yeah, so ideally, for any multi-center trial, you should be looking at analyzing, including center, and then looking at center-specific effects as well, because Sam's right. Certainly, you can get variation from site to site, and that may be allowed in the protocol, or you may want to shore up that area in the protocol, and that's why having all this formative work in talking with hematologists and talking with patients helps, because it helps you understand those sort of local nuances, and whether in your protocol that you want to allow for that or be very narrow is important.
Great. Thanks, everyone. This is the end of the Q&A session, but we still have another 15 or so minutes left on the webinar. If you have any more questions, feel free to continue using the Q&A, and we'll do our best to answer them directly in there, or indeed reach out to any of the speakers offline as well. I'm sure they'd be happy to answer your questions. And I'll hand it back over to Megan.
Yeah, and Justin, thank you for saying that. So we are now moving into another 15 minutes that are Q&A. They are meant for stakeholders who may wish to ask more plain language type questions, either about the research or perhaps the patient perspective from Terry and his engagement in research. So we'll do priority for those questions. However, I think if the rest of the panelists are willing to stay on, we'd be glad to answer other questions as well. Before we begin that, I would like to make a couple of comments. So the first being, thank you to all of the panelists. I just can't imagine how you made it possible to fit so much information in such a succinct way. It was really, really excellent, and it was quite a story. And then secondly, more from a housekeeping point of view, at the end of the session, you will, as participants, be receiving a link to a survey, and we do really ask that you fill that out, because that's helping inform how we do these sessions. So with that being said, Justin, perhaps I'll pass it over to you, and maybe just for sake of being able to see you guys a little bit better, if you don't mind stopping your screen sharing, then we can kind of just see you guys bigger and better. There we go, perfect. And again, the next part of the session will end at 3:15. So Justin, back to you.
Sure. So again, any questions from anyone listening in for Terry in particular, or Madison, or anyone else? And while you're thinking, I guess taking moderator's prerogative, a question to you, Terry, if there's anyone on the webinar with lived experience here that is sort of interested, maybe excited at the prospect of partnering in these types of programs, but maybe feeling a bit apprehensive given everything that we've talked about, where do you think they should start, or what are the first few steps do you think they should take?
Yeah, that's a great question. I'll just kind of relate my own experience, and I had a burning desire to want to just get engaged in healthcare and be able to advocate patient healthcare issues, but I really didn't know where to begin. And where I did begin, I got some advice from an individual at the Leukemia Lymphoma Society of Canada, where to start looking to really self-educate myself. And there's I would really start with using some of the wonderful resources that are available via the internet. Specifically, there's the SPORE strategy for patient-oriented research website has some great information about patient engagement, as does the American equivalent, PCORI, the British equivalent, Involve. All of them have some wonderful resources about what patient engagement is about, and how, you know, and how to kind of get started in it. So that's where I would start. And you know, there also are some I think some formal training programs online that you can tap into if you're involved or associated with a patient group of some sort, like myself, the Leukemia and Lymphoma Society of Canada. You can go to them, and they may have one or two individuals that can help point you in the right direction as well. So that's kind of, you know, I guess my approach.
Brilliant, thanks. I'm just noting Megan posted something also saying that you can get involved in the BioCanRx Learning Institute, and there's some links there as well.
Yeah.
Fantastic. Still not seeing any additional, don't be shy if you have any questions for clarification.
Justin, I'm not sure if you can see it, but Sandra Donaldson asked a question. For randomized trials, will the statistical analyses be stratified by center due to variability of the therapy, or has this become more standardized?
I think Dean covered some of that, but he might want to expand on it.
Okay, I'm sorry. Yep.
Any other? I mean, I've got plenty of questions, so I can keep going if no one wants to to pipe in. So I guess a question for Kednapa and Mackenzie, possibly around just involving patients and people with lived experience in not only economic evaluation but early economic evaluation. And in principle, that doesn't seem to be happening, is what you said. And some of the work that you're doing is enabling some of that to happen. And I'm just interested in, you know, how have patients and people with lived experience here found that experience? Have you found it, they've been able to, or are happy to engage? Is it too complex? Is it easier than you thought? Any thoughts on that?
Mackenzie, do you mind take this question, the fact that you're heavily involved in that stage? Thank you.
I'm happy to speak to that. So again, as I mentioned, as Terry referenced as well, we've only hosted 2 discussions so far, but we are striving to engage beyond patients and caregivers, also healthcare professionals and policymakers and payers. And out of those 3 stakeholder groups, we have gotten the greatest response from patients and their caregivers. So I think that is one kind of signaling factor that there definitely is an interest in this, and that folks do want to be engaged, just based on the response that we are getting. And also we are evaluating this form of engagement, as I said, to inform ultimately a framework down the line. And the feedback we are getting is that folks have appreciated the opportunity to engage. And obviously, because of the pandemic, this engagement it looked different than what we had planned initially. So there were some hurdles, as Madison showcased at the beginning of our talk this afternoon, that we had to kind of navigate. So these discussions are happening online, which I think has provided the opportunity for folks to engage from kind of a wider geographical spread across the country. So we have had folks participating from different provinces, and I don't think any territories, but yeah, so engagement has been great in terms of patients and caregivers. And now we are just kind of working to kind of think about ways and methods that we can tap into those other stakeholder groups.
Yeah. And on that note, I just want to advertise our project here, Mackenzie. The fact that right now we actually moving toward 2 other stakeholder groups. So if you're interested in joining our project, like if you are healthcare payers, if you are clinicians, feel free to contact myself or Mackenzie. So we'll reach out to you, and it's going to be fun.
Yeah, absolutely.
Brilliant. Still no questions. Don't be shy. Maybe one for Josh. fascinating to move from the sort of standard reviews and meta-analysis to individual patient level. And I'm just wondering if you can speak a bit to, I guess you'd need to get access to the data sets eventually, and how do you envisage that? Have you already started to reach out to those studies that are already in the existing systematic review? And going forward, how would you encourage people to contribute to that in order to enable the analysis to happen?
Yeah, thanks, Justin. It's a good question. When we came up with the idea originally, we did sort of, I guess, called it run a pilot, and we looked at the 42 studies that we had included in our review originally, and actually 35 of them had patient level data. So a lot of them were small, sort of 4, 5, 6, patient trials, which provide that granular data. And the ones that didn't were kind of larger, Juliet trials from Novartis and stuff like that. So I get the regular channel would be to contact the primary author or the corresponding author of the trial and try to get the data. But so that'll be what we'll do. I'm not holding out hope that these large drug companies are going to just provide us with their individual patient data, in which case we'll simply include them in their review. But their participants, their patients that are included in the study, just won't be able to be included in our analysis. But we'll do everything we can to try to get that data. But yeah, especially for the larger industry funded stuff, we've tried to get similar data for other studies, and we've hit a wall a few times.
Hopefully the move towards more open science practices is going to help towards doing that. And the fact that you're updating the reviews continuously kind of puts the team on the map around, this is going to come down the line if you're planning the next trial that have your data ready for us.
Yeah, hopefully.
I had a question for Terry, if that's okay. Terry, I was just going to say that I really enjoyed working on these projects, and I know it's definitely been a learning experience for me, and I've learned a lot. And it's been great and really eye-opening to hear more about your perspectives. And I guess I was just interested in if there was kind of an activity that you enjoyed most when you were working on throughout the GoCart program.
Well, that the meeting we had, there were some great sandwiches that are just wonderful. No, I can't really pin anything down to one specific activity other than in a broader sense, the learning experience for me has been incredible. I've now been able to see things from the other side, from the researchers side, and they're good people. They're not evil. They're really out for our best interests. And just the understanding of gaining the understanding of the research process has been so good. And I have to say that it's really been an encouraging experience and a fulfilling experience. And I'd have to say the support from all the individuals and the mentorship has been greatly appreciated. So I can't really pick out one specific thing, but it's just in the broader sense, the whole experience has been fantastic, and it's largely because of the people that you're dealing with, and you're all good folks, and your heads are in the right direction. Is that a good answer?
Yes, no, thank you so much, Terry.
I do have a question. Maybe I'll direct this to Justin or Dean or Manoj. Describe your ideal patient partner in research, and what does successful patient engagement look like to you?
Great question. So for me, well, I'll preface it by saying that the projects that research projects that I've been involved in where patients have been involved have been incredible. Just hearing experiences, integrating that knowledge into the projects and into the findings and into the dissemination of those findings. It's incredibly rewarding from a researcher's perspective. In terms of the sort of ideal patients, it's those that are clearly can speak their mind, are not afraid to say anything, and that work well within a team environment. Not just who those team members are, but just that respect that obviously we all share, and it just leads to a great environment where people are free to speak their mind, and are respectful. And we've been so fortunate to have that with the vast majority of our projects, and obviously that respect is going back, that we have to create that environment, and yeah, but there's been very few that I where it hasn't worked. Quite frankly, there's more researchers that you're more worried about than the patients, to be frank. Yeah, it's a great question. Manoj or Justin?
Yeah, no, I'd agree with what you said, Dean. And like in my mind, certainly for some of the projects we've been doing lately, especially, there is no precedent for patient engagement, for instance, or very few precedents, I would say, for patient engagement in laboratory research, right? Which is something that we've been moving into as well. So in that case, and other things we're doing, I think having patient partners who recognize that this will be sort of exploratory work for everyone involved, and trying to figure out together how this might all look is very important as well too. And there's certainly a piece of that working with Terry and Stu for this for the CAR program as well, because that was, for myself as a researcher, one of my first forays into that. So having people who recognize that we won't have all the answers as to how this is all going to work is important as well too. And I think in terms of background, there's one last point I just say is that, I think diversity is important, and part of the struggle has been to find an adequate number of patient partners for many of our programs. So, we'd welcome everyone basically almost, right, that's interested in research, and we can probably find something that we can work on together.
Yeah, I'd echo that. I think for me, an ideal patient partner is, it's hard to define one, because I think the ideal is to have many different perspectives, and anything that we can do to work towards that, it's always a good thing. And I think adding to what makes a successful partnership, I think for me, it's a partnership that has mutual benefits. So not only that the project team gains from having someone with lived experience being partnered, but that the patient partner, someone with lived experience, also benefits from contributing their lived experience, but also potentially contributing their professional or other skill sets, and there may be additional ways beyond their patient role that they can contribute to the greater project and benefit from upskilling as well. So mutual benefit, I think, is an indicator of success for me.
Thank you, Justin. So I think that concludes our session. And again, I just want to thank you all for participating, and for the participants for joining this session. As I mentioned, we are having our third session. We'll be hosting that in the new year, in January. And so I invite you all to participate, and you'll be hearing more about that session either through our newsletter or via social media. So until then, I wish everyone a happy new year, and take care, everyone. Goodbye.
Bye.